CRISPR operates as an ancient bacterial immune defense system. When an invading virus attacks, a specialized enzyme intercepts and neutralizes the viral strand by cutting its DNA in half. What began as an obscure observation of prokaryotic immunity in 1987 transitioned in 2012 into a fully programmable genome editing platform. Looking at this single green plant surviving in a cracked, desolate field, we see the first high-stakes application of that transition, engineering climate-resilient agriculture to secure global food supplies against extreme weather. These orange-brown spheres are SARS-CoV-2 particles. Here, CRISPR is deployed to provide rapid, high-precision infectious disease diagnostics, capable of identifying viral RNA during pandemic scenarios. This is the United States Patent and Trademark Office, the staging ground for a multi-billion-dollar intellectual property war over the underlying rights to this technology. The legal battle pits the Broad Institute against a formidable coalition represented by the University of California and the University of Vienna, known as CVC. To understand how a single biological mechanism commands such commercial dominance and poses such complex ethical questions, we have to deconstruct the specific cleavage properties of the different Cas enzymes. The baseline mechanism of CRISPR relies on two components working in tandem: a guide RNA that searches for a specific genetic sequence, and a Cas protein that binds and cuts the target. This table maps out the cleavage patterns. Cas9 targets double-stranded DNA and creates a blunt end cut. To execute this, it strictly requires a G-rich sequence called a protospacer adjacent motif, or PAM, located directly next to the target site. Cas12a also targets double-stranded DNA, but its profile differs. It produces staggered end cuts, leaving overlapping tails of DNA, and requires a T-rich PAM to initiate the cut. Cas13 ignores DNA entirely. It targets single-stranded RNA, relying on a protospacer flanking site to bind to the sequence and trigger RNA degradation. Then there is dead Cas9, or dCas9. This is an engineered variant that binds securely to double-stranded DNA without cleaving it at all. Because dCas9 anchors to a specific sequence without breaking the helix, researchers fuse it with transcriptional effectors. This creates CRISPR interference to turn genes off, or CRISPR activation to turn them on, allowing for reversible epigenetic editing. The selection of the Cas nuclease dictates the biological outcome. The specific enzyme determines whether a scientist will knock out a genome, destroy a circulating virus, or modulate epigenetic expression without altering the underlying code. A Cas9 double-stranded break is biologically destructive on its own. To make it useful, biotechnologists have to manipulate the cell's endogenous DNA repair pathways immediately after the cut is made. The first pathway is non-homologous end joining, or NHEJ. In this error-prone process, the cell sloppily stitches the broken ends back together. By hijacking this repair, scientists reliably create mutations that disable or knock out specific genes. The alternative is homology-directed repair, or HDR. Here, researchers supply a donor template alongside the Cas enzyme. The cell uses this template as a blueprint, flawlessly integrating a new genetic sequence to achieve precise knock-ins. These molecular repair pathways scale far beyond laboratory cell cultures. They are applied to engineer climate resilience directly into macroscopic agricultural crops. For example, researchers used Cas9 to knock out the SL-MAPK3 gene in tomatoes. Disabling this single target allowed them to map the resulting transcriptional cascade and isolate the mechanisms controlling drought resistance. A similar strategy was applied to rice. Scientists targeted the OSPYL9 abscisic acid receptor gene, mutating it to alter the plant's natural stress response pathways. Looking at this golden rice field, we see the physiological outcome of that OSPY9 edit. Under severe water-scarce conditions, the modified plants achieved a 25% increase in grain yield. This approach carries a strict regulatory advantage. By utilizing CRISPR ribonucleoproteins, scientists produce edited crops that contain zero foreign transgenic material, classifying them as DNA-free. Mastering cellular DNA repair pathways enables biotechnologists to secure global food chains without relying on the heavily regulated techniques used for traditional genetically modified organisms. While Cas9 is optimized for agricultural DNA, the Cas13 nuclease is deployed to target the single-stranded RNA of circulating viruses. Cas13 exhibits a biological anomaly known as collateral cleavage. Once the enzyme successfully binds to its target viral RNA, it changes shape, becoming a highly active, non-specific RNA-degrading machine. Scientists exploit this collateral degradation by flooding the sample with fluorescent reporter molecules. When Cas13 activates and begins shredding everything in its vicinity, it cuts these molecules, releasing a measurable burst of fluorescence. That fluorescent release forms the mechanical basis for microfluidic CRISPR diagnostic platforms, including systems like Sherlock and Detector. These rapid diagnostic platforms are programmed to identify the specific genomic signatures of severe viral threats, including the SARS-CoV-2 virus, the Ebola virus, and influenza strains A and B. The logistical advantage is significant. Triggered simply by RNA binding and collateral cleavage, these platforms deliver point-of-care, highly specific viral detection without the need for complex, expensive laboratory equipment. By exploiting an enzyme's off-target collateral degradation, researchers transformed a gene-editing mechanism into a highly mobile pathogen surveillance network. Moving from test tubes into human medicine requires crossing a biological barrier, plunging through a dense cell membrane, navigating the cytoplasm, and piercing the nucleus to reach complex chromatin demonstrates the hostility of the eukaryotic cell architecture. That cellular barrier sits at the exact center of the Patent Trial and Appeal Board interference dispute between the Broad Institute and CVC. In this image, Jennifer Doudna holds her 2020 Nobel Prize certificate, recognizing her role as a primary inventor for the CVC team in detailing the Cas9 cleavage mechanism. Beside her in that effort was Emmanuelle Charpentier, seen here holding her own Nobel medal. Together, they formed the core of the CVC coalition, whose research defined the biochemistry of CRISPR-Cas9. On the opposing side is Feng Zhang, presenting his research in this image. Operating out of the Broad Institute, Zhang led the team that executed the first successful CRISPR edits inside mammalian cells. The legal threshold debated at the patent board came down to a strict difference between the theoretical conception of an idea and its actual reduction to practice. CVC's legal argument rested on the timeline. They proved they were the first to demonstrate Cas9 DNA cleavage on purified DNA in an in vitro test tube assay. Broad's counter-argument focused on execution. They maintained that proving cleavage in a sterile test tube does not prove the Cas9 system can successfully navigate eukaryotic chromatin and hijack repair pathways inside a living cell. The patent board ruled in favor of Broad. They affirmed that adapting CRISPR for the hostile environment of eukaryotic cells was not an obvious extension of the test tube data, but a separate, patentably distinct invention. In the eyes of intellectual property law, the sheer biological complexity of the eukaryotic nucleus makes theoretical in vitro conception completely distinct from functional in vivo execution. Beyond legal thresholds lies an ultimate ethical boundary, deciding how and when to edit the human genome to treat intractable diseases. Current clinical applications focus entirely on somatic gene editing. This involves modifying non-heritable cells to treat a specific living patient. We see this in active clinical trials where scientists are knocking out PD-CD1 and ACE2 receptors in CD8-positive T cells. This prevents viral entry and stops T cell exhaustion, all without affecting the patient's offspring. This widely accepted therapy contrasts sharply with germline editing, the modification of embryos or gametes in a way that passes genetic alterations down to descendants. This photograph captures He Jiankui at a genome editing summit, shortly after his premature experiments editing human embryos caused an ethical flashpoint, eroding public trust. The risks of germline edits are profound. They force changes on unconsenting generations, carry dangers of off-target effects, and open doors to genetic enhancement. In response, bodies like the World Health Organization, seen here, and the Third International Summit established a firm consensus. Heritable human genome editing remains unequivocally unacceptable until rigorous safety standards are met. The programmable precision of the Cas nucleus suite allows us to secure crops, build rapid diagnostics, and target intractable diseases. But the permanent alteration of human inheritance remains a strict ethical boundary the global community refuses to cross.